Yeah, I would like to start with this quote. And I would like to convince you in my talk that René Descartes was able to make this quote and to say, I think, therefore I am, and we all think, and therefore we are, because our gut allows us to do, because our gut has a mind of its own, because our gut has its own brain. And because our gut takes care of its own business, it allows us to think and don't think about digestion and all these kind of things, which are important as well to survive. Anyway, I hope I convince you after my talk with this. So today I'm going to talk about gut-brain axis alterations in a mouse model for neurodevelopmental disorders, which is sort of a sidestep we took in the lab with one PhD student. And before we really dig into this part of my talk, I would like to introduce you to the enteric nervous system, which is this brain of the gut, of course, which I was alluded to in my new opening slide. I think it's really important that you guys who are in this field of gut-brain axis and brain-body interactions are aware of the enteric nervous system for the reasons I just mentioned. Because I think it's really an important part of the gut-brain axis. It's sort of a relay station located in the gut that is influenced also by all the molecules and signaling pathways that have been discussed. But it's also an important nervous system that is affected not only in gastrointestinal disorders, but also in a lot of brain disorders. And so for that reason, effects on the enteric nervous system might not be a cause of, let's say, Parkinson's disease or Alzheimer's disease, but a lot of the patients with these type of disorders, also patients with mental disorders, neurodevelopmental disorders, have GI symptoms. That can be a brain-to-gut effect, but it can also be caused by simple pathophysiological mechanisms that happen in the nervous system within the gut wall. And this is something that we are interested in the lab, and this is how we ended up also looking into neurodevelopmental disorders. So the enteric nervous system is a nervous system located in the gut wall, the whole length of the gut, upper way from the esophagus, all the way down to the distal parts of the rectum. In the gut wall, generally speaking, this enteric nervous system is organized in two plexus layers. The outside myenteric plexus layer, which is located between the longitudinal and the circular muscle layer, and the submucous plexus underneath the mucosa. If we take a look under the microscope at the ganglia of the myenteric plexus, you see this network, which are actually connected ganglia in which neuronal cell bodies are located, and these are intermingled with supportive enteric glial cells like astrocytes in the brain, so we call them enteric glia. And this network contains different types of neurons that regulate gut functions independently from input from the other parts of the autonomic nervous system. That doesn't mean it's not influenced by those parasympathetic and sympathetic innervation, but it can do stuff autonomously. And that is shown here. So what you see in this lower movie is an isolated large intestine of the mouse, which I sacrificed, unfortunately, put the large intestine in an organ bath and put a rice grain in the proximal part of the colon, that is. And you can see that with all connections to the brain spinal cord disrupted, just a piece of gut in an organ bath, the gut is still able to propel this pellet distally, which is this process called peristalsis. And that is because of the presence of this enteric nervous system. So if you look at the schematic here on top, again, you have the MP myenteric plexus and SMP submucosal plexus. So the neurons within this myenteric plexus, they're located in between the muscle layers. Their main function is to control contraction and relaxation of those muscles, so control something like peristalsis. And then the cells within the submucous plexus are underneath the mucosa, and they control submucosal plexus things, mucosal stuff, absorption, secretion, local blood flow, these kind of things. So this is what the ENS is doing. Again, we use a lot of microscopy, fluorescence microscopy, mouse work. Up till now, most people were talking about human research, and mouse work and rodent work is criticized, but still it's our favorite model. So I'll be talking about mice. So what you see here are pictures taken with the confocal microscope of the myenteric plexus and submucosal plexus. And you see that these neurons, here labeled with GFP or in green YFP, are in these ganglia, and the SOX10 glia is a transcription factor that we use to label the nuclei of these supportive glial cells. They're situated in between. And what is interesting is that while the neurons are located exclusively in those plexus layers, those glial cells are actually present in all layers throughout the gut. So they also find them at the mucosal crypts and in the very tips, in the tips of the mucosal villi, which is important for actually Gabriele's talk will present after the lunch break. So please remind that. So one of the reasons why we study the enteric nervous system, because it's beautiful. What you see here is a recording from Gabriele as well, from light sheet microscopy. So here we use a transgenic mouse model in which the enteric glial cells are labeled with a fluorescent reporter, TD tomato, which is red fluorescent. This piece of intestine, colon in this case, was cleared and then put under a light sheet microscope. And if I play the movie, you will see this vast, extensive network that is located in the gut wall. This is mostly what you see is my enteric plexus, but if we zoom in, you will also see submucosal ganglia of the enteric glial cell. So this is the whole network. If you make transversal section in the next slide of the colon, this is also an image I took from Gabriele's work in the colon, and you see the glial cells. So you see my enteric plexus, submucosal plexus at the outside, and then you see those glial cells, these mucosal enteric glial cells that are present in the mucosal folds. Yeah, we're really interested in those cells, and that is Gabriele's project, so I won't talk about that today. Anyway, the ENS is beautiful. Another reason why we study the enteric nervous system is because it's way more complex than the first brain, than the CNS. I'll try to convince you. On the left, you have the second brain, so the enteric nervous system, and you see a collection of ganglia, and you see all different types of enteric neurons. Here labeled with a different color. So we have excitatory neurons, inhibitory neurons, and then subtypes of those. They're all intermingled within these ganglia. But there doesn't seem to be any logic order of how they are grouped within these ganglia. If you look at this first brain, this is an image of the mouse cortex where different types of interneurons are again labeled with transgenic reporters. It's organized in layers. So if you go to a particular brain layer, you will find a particular subtype. That's easy, right? You can just go to the brain or look to this layer and find this cell type. In the gut, it's chaos. It's really difficult to make sense of this very chaotic network of all these subtypes of neurons. So that's a reason why we also are interested and intrigued by the second brain. And eventually what we would like to do, of course, is to draw up a map of the enteric nervous system to see how all these cells are plugged in. And of course, this is the map of the tube map of London, which is actually where I started, or this research, which I started as a postdoc in London, and I still use this map. It's changed over the time, so I should actually update it. But at least that's the goal. So, what are the tools that we use in our lab to understand this enteric nervous system? We use a lot of microscopy and transgenic mouse lines. So on the left, for example, you see sparse labeling technology to look at the morphology of these individual enteric glial cells. What you see here in the movie is a calcium imaging recording of activity of enteric neurons and glial cells in the submucosal plexus. These are images similar to the light sheet I showed before. We use transcriptomics, profiling of microRNAs. We use a lot of gut function assays, transit time measurements, so we gavage a dye into the stomach of the mouse and then wait for the first colored poop to come out, and that's a time measurement of the whole transit time, so an EC measurement. We make a permeability assay and stuff like that. We also work with flatworms, Schmittea mediterranea, and what you see in this image here is again a confocal image, is actually the gut of that flatworm, which are very remarkable species, and they have innervation in the gut, and they are actually like the masters of regeneration. So if you cut this worm in half, it will completely make 2 new worms. And we believe that because the gut is also regenerating constantly, or epithelial cells are, and we think there is a role for the nervous system, and that's why we moved to this flatworm to see if we can cut the worm in 2 to see if also the gut is regenerating and how the nervous system is involved in that. So a couple of slides on the enteric nervous system, things that I thought were relevant for you guys. The enteric nervous system is there to control gastrointestinal function independently from the central nervous system. So, for example, what it does, and this is a study that was performed mainly in Leuven, where I did my PhD, and we're still collaborating with the lab there. It's a paper that was already in bioRxiv from 2021, but got recently accepted in Nature and will be online next week. So what we found actually is that the enteric nervous system is sensitive to nutrients, specific nutrients. For example, here are glucose and acetate, and depending on the type of nutrient that you spritz onto the lumen, onto the mucosal surface, different neurons in the enteric nervous system will become activated. This goes via activation of androchromaffin cells that release serotonin, and the model sort of is like looking like this, where these nutrients activate EECs, androchromaffin cells, that release serotonin and then activate descending pathways and ascending pathways in the enteric nervous system to make sure that nutrient absorption is integrated with motility. That, okay, if I have nutrients here, I need to push forward the bolus, or I need to make segmenting contractions and these kind of things. So this computational work is performed by these circuits integrated into the gut wall. Another thing I would like to show is how the ENS is actually adapted to its local environment. So here, this is RNA sequencing data, single nuclei sequencing data from our collaborators in London, comparing the transcriptome of enteric neurons in the small intestine versus the colon. And we know, we've talked about microbiota. Microbial diversity is way more diverse, and there's higher loads in the colon. And this sort of corresponds with some of the genes that are upregulated in the colon, in enteric neurons of the colon. So what Yuki did was actually compare, again, germ-free with SPF mice in the colon. And you see that some particular genes are upregulated in germ-free mice in enteric neurons of the colon. So microbial colonization inflicts changes onto those enteric neurons. And one of the important transcription factors that you see upregulated is, for example, aryl hydrocarbon receptor, the AHR receptor, which is a transcription factor that sort of acts as a biosensor for microbiota signaling and adapts the enteric nervous system to the presence of different types of bacteria, which is activated by certain types of short-chain fatty acids that Lucas was alluding to. So the ENS is, of course, important for homeostasis, but is also involved in a lot of gastrointestinal disorders. We've heard about IBS before. There's also inflammatory bowel disorders. There's even colorectal cancer, where the ENS maybe is not at the origin of the disorder, but is definitely affected. And then there's, of course, all the central nervous system disorders. But there's a group of disorders called enteric neuropathies, where the cause of the disease is really a malfunctioning or an error in the enteric nervous system. And the prototype of the disorder is called Hirschsprung disease, where there is actually in the distal part of the colon a lack of an enteric nervous system. And this usually is because of developmental deficits during embryonic development of how the enteric nervous system develops. 1 in 5,000 live births, so the ENS is absent in the distal gut. And survival of these children really depends on early diagnosis and surgical intervention. So the part of the gut where there's no enteric nervous system is removed. So there's a lot of research in our field trying to understand this disorder, but it's also an interesting field to look into just to understand how the enteric nervous system develops. So of course we use mice to study this. This is an example of the EDNRB knockout mouse, which is an important receptor for ENS development. If you knock that out, you actually create in the mouse sort of a Hirschsprung phenotype. So that means in the distal part of the colon, there is a part where there is no ganglia of the myenteric plexus, so no enteric nervous system. This is how the proximal colon looks like on the left, normal. A transition zone where there's lower amounts of ganglia, and then an aganglionic zone where there's no ganglia anymore. You still see fibers there. Those are from the extrinsic innervation, but you also still see the glia there, which are here labeled in green, which we think are an interesting cell to target for this disease. What I would also like to convince you about is that I think I'm convinced that there are disorders, mostly GI disorders, where subtle defects in the enteric nervous system are actually the cause of the disease. So Hirschsprung is like the ENS is absent in a specific part of the gut, and that causes the disease. But I think very subtle changes in the network, like we have in the brain for specific diseases, can also be a cause of GI disease, not just absence of cells or a lower number of ganglia. And this is something that we studied already quite a while ago, so I'll try to explain the model. So we're using, again, mouse transgenic lines. And what we've done in this experiment is to create a mouse line with a deletion of Salazar-3, and this is a gene which is important for orientation of cells. It was actually identified first in the fruit fly wing for the orientation of the cells for the polarity in the wing. So we found it to be expressed in developing enteric neurons. So we created a mouse line where during development, the enteric neurons, progenitors, lack this molecule. And what happens actually is that during development, early stages, the cells usually grow into the gut, they start to make the network, and they send out their axons. And usually they send out their axons distally. Now, if we delete Salazar-3 and enteric neurons, there's this randomization of projections. So normally the predominant projection is oral to anal of the axon, but you see in the knockouts, this is more sort of messed up. Also, the processes are shorter. These mice actually look pretty normal. They're a bit smaller, but they survive. But if we look at their intestines, the ENS is there, all the cells are there. It's just that the cables are a bit unplugged incorrectly. So if we look at gut function, we see that transit time is delayed here. The poops are smaller, the fecal pellets are smaller, and we make these movies. And here you see the difference between the control and the mutant line. So the control line is actually the same movie as I showed in the beginning. This is rice grain going distally with peristalsis. In the mutants, the rice grain gets stuck. Eventually, some of the mice were able to propel the pellet, but what happens here is because of this misprojection of these enteric neurons, there's actually contractions happening on the opposite side of the pellet, where there should be relaxation on the distal side and a contraction on the oral side to push the pellet distally. Peristalsis. This now is like interrupted and it gets stuck. I really think that these subtle changes in network of the enteric nervous system connectivity can be a cause of several GI disorders, but it has to be proven, of course, in humans. So I started my talk with trying to convince you that the ENS is something we should not forget in the gut-brain axis, for which I'm not an expert, but we sort of accidentally ended up in this field. But I think the ENS is really a relay station along the gut-brain axis because it is connected with the parasympathetic and the sympathetic nervous system. And it's really, like I said in the beginning, it is influenced by microbiota. It is influenced by nutrients, which then can be, these signals can be transmitted to other parts of the nervous system or to the bloodstream. So what is the role of the enteric nervous system in CNS disorders? There's a lot of research, for example, in Parkinson's disease, where people think that Parkinson's disease, or there's theories that Parkinson's disease might even arise in the intestine. Also, there's theories in Alzheimer's disease, but what is definitely sure is that all these patients with these disorders, or a lot of them, suffer from gastrointestinal complaints. So the very simple question that we took is, how is the ENS affected in these disorders? If they have constipation in Parkinson's disease, for example, is that because of some brain to gut signaling, or is it just because the network in the gut wall itself is just affected with same pathophysiological mechanisms that are occurring in the brain? So this is work mainly driven by Kinga, a PhD student who still has to defend, but she already finished her 5 years. But Kinga started with looking into Alzheimer's disease. So for that, we use a mouse line. This is the 5x fat model of AD. So this mouse line overexpresses two key human proteins with mutations related to familial Alzheimer's disease in APP, so amyloid precursor protein, and in presenilin. And these two mutations are driven by the Taiwan promoter. So the Taiwan promoter is a promoter that is active in neurons to make sure that these mutations are driven, or overexpression of these mutations is driven in neurons. This was designed to induce these mutations in the brain, of course, so we look at the ENS. Important, this model recapitulates many cognitive and pathological changes observed in Alzheimer's. We can talk about the translation later, but at least it's a very common and very useful model in the fundamental field of Alzheimer's disease research. So what Kinga actually first found is that APP, the transgene, is expressed in the intestine. It is very much upregulated compared to wild-type animals. So this Taiwan promoter is also active in neurons in the gut. So it drives the mutation of those proteins in the gut, both in the small intestine and in the colon. And if we look through the microscope at those enteric neurons, and we use antibodies for amyloid, beta amyloid, we find accumulation of beta amyloid in enteric neurons in those 5x fat mice, which is sort of not amyloid plaques that you find in patients with Alzheimer's disease at later stages, but at least it's sort of the start of the pathology that's also happening in the brain. So what does it mean for the enteric nervous system? Actually, we don't see major changes in terms of numbers of enteric neurons or subtypes of enteric neurons, but these mice have altered gut function. They have actually faster intestinal transit in this model, which is not corresponding with some of the features that people find in patients, which is predominantly constipation. So, but there is something happening in the gut of these animals. The stools of those mice are bigger, and there is a change in their water content. Importantly, what we also find in these animals is that they have low-grade intestinal inflammation. So by overexpressing a transgene in neurons, these mice develop sort of mild inflammation. There's an accumulation of inflammatory cells in the gut wall, signs of intestinal fibrosis, enlarged and activators, Peyer's patches, and regulation of inflammatory cytokines, just by inducing overexpression of this transgene in enteric neurons. So yeah, this work sort of highlights the importance, at least I think, of peripheral pathology in central nervous system disorders, whether this is a cause or contributes somehow to Alzheimer's disease pathology in the CNS, I would say, but at least I think we can explain using these type of models why people with these disorders get GI symptoms. For me, it's more logic that this is happening like peripheral pathology than at least not only a contribution from brain to gut. So now I get to the title of my talk, which is neurodevelopmental disorders. So this is actually a collaboration with a group of in biomed of Werend, who's looking at the central nervous system and focuses on microglia. And he's very interested in neurodevelopmental disorders. And he uses a mouse line, again, a mouse line, which are called DISC1 locus impaired mice. So DISC1 is a gene that was discovered in 2000 in a Scottish pedigree, and those patients were suffering with severe mental disorders. Several people in the family had schizophrenia, autism spectrum disorder was reported and stuff like that. So collaborators of my collaborator in Hasselt developed a mouse line where there's a locus impairment of DISC1, which is actually inducing a knockdown malfunction of this gene. And this gene this protein is very important for nervous system development. It's really an important developmental regulator, important for neuronal maturation and connectivity of the central nervous system networks. So in this mouse model, these locus impaired mice are smaller, but they have structural alterations in the brain in terms of the circuitry and the network at synaptic level, but they have severe behavioral and cognitive deficits. And here you can see some of the results of behavioral assays that were performed in the study. So, of course, we are interested in the gut, so that's what we did. So first we checked whether this DISC1 gene is actually expressed in the enteric nervous system, and with RNA-scope, so in situ hybridization, looking at the mRNA of DISC1, we find that it's overexpressed, that is expressed in enteric glial cells, and in the knockout animals, there is a clear reduction of DISC1 in the gut wall. So, and that is also here validated with qPCR at the mRNA expression level. So, in terms of body weight, these mice are a bit smaller, their intestines are shorter, because what we found in the Alzheimer's disease model, we also checked with histology whether we could see signs of inflammation, but we didn't. So actually the gut wall looked sort of normal. There was no signs of inflammation or upregulation of inflammatory cytokines. But again, the mice had different gastrointestinal transit time. And here we see, again, faster gastrointestinal transit time in the locus impaired animals. And because the mice are smaller and the guts are shorter, we correct for that and we measure the velocity, and you see that the transit time is actually still faster in those mouse lines. Now, again, looking at the enteric nervous system, here we do see a higher number of enteric neurons in these mutant animals. We don't see changes in the different subtypes, and actually this is still ongoing work. I had to say it from the beginning, but we find an increase in the number of enteric neurons. I actually really don't know what this means. It probably is a developmental defect. It has been reported in some models that there are immature neurons surviving in later stages, and it could be something like this is something that we try to understand. Well, now we looked at intestinal microbiota. I'm not an expert. These slides should say that alpha and beta diversity are not majorly affected in these mice, but we see here a significant effect on phage phylogenetic diversity. I'll quickly move to this slide, which is easier for me. If we do linear discriminant analysis, we see that there is an upregulation or a higher abundance of Bacteroides species in the locus impaired animals, and Clostridia are almost absent. It was alluded before by you, Lucas, that these are actually found to be also higher abundant in people with depression, right?
The Bacteroides two enterotype. I mean, there's two Bacteroides predominant enterotypes, but indeed, it's Bacteroides.
Yeah. So for what it's worth, translational, at least we find these. This is rodent stuff and rodent data, but for me it's really spectacular that we find differences within these locus impaired animals. You have to imagine that they are housed together. So mutant animals are living in the same cage as wild type littermates. The only difference that they have is a mutation in this gene. They eat each other's poop all the time. Still, we find this, which I find very striking. So that said, okay, there must be a role or an effect of microbiota. So obviously, the next thing to do is do fecal microbiota transplantation. And so this is like the study that we performed. We're still suffering low power. We still have to repeat this, but I'll show you the preliminary data. So we give mutant animals species from wild types, and we do also the opposite. We give the mutants. We give the wild type animals species of the mutants, or is that what I already said? Okay, we know what I see here. So for 3 weeks, and then we start with gut function assays and also behavioral assays. We don't see effects on gut function after FMT. This is important, I think, because it means that the deficiency that we found, or the effect on transit time we found in the mutants, is probably because of a local mutation, a local effect on the enteric nervous system, and here the contribution of the microbiota is maybe not that important for changing gut function, which I think is important. But then we also move to behavioral assays, still suffering low power, but this object location task, which is a hippocampus-driven cognitive task, is rescued by giving locus impaired animals wild type microbiota. That is spectacular. But of course, we're interested in what the mechanism is and how does it work. So this is something that we are trying to pursue. I mean, we've seen human studies even with microbiota transplantation being very effective. But how is this working? So Werend, my colleague at UHasselt, looks into microglia, and he knows that he's found that those microglia in those disc-1 locus-impaired animals are deficient. These animals have hyperphagocytic microglia. They engulf more synapses early on during development, which is important for making the CNS network. They also in vitro, these microglia, locus-impaired microglia, take up more synaptosomes. And in the brain, this leads to impaired neurotransmission in the hippocampus, and also a decrease in the number of synapses in the hippocampus, because those locus-impaired microglia are hyperactive. They phagocytize too much. Werend's group could rescue this phenotype by bone marrow transplanting wild-type microglia into the locus-impaired animals. So obviously my question is now, what is the effect of the microbiota treatment on those microglia? Can we rescue the phenotype? Do we rescue the phenotype at the behavioral level because we change the microglial function? And of course, that's what we are hoping to show in the future. Unfortunately, that's not what we're doing yet. But maybe next year at that multi-coast I can show. So yeah, this one phenotype is rescued by bone marrow transplant of wild-type microglia, but obviously we think, or we hypothesize, that microbiota changes might also change the phenotype of those microglia. With that, I would like to thank everybody in my lab and you for listening, of course also the collaborators and the people for fun. Thanks for
you. So we have time for some questions.
Yeah, because very nice talk. I agree completely with you that the enteric nervous system is an important relay station in the microbiological brain axis. I mean, no doubt. I think the problem is, of course, I mean, and we've discussed this oftentimes. But
not always out in the public. Now we do.
Yeah, well, yeah, mostly in some kind of dark bar. But yeah, the problem is just like we did the whole discussion earlier about the afferent vagus. It's extremely hard to measure in humans, right? I mean, we may not find the solution, but I think we have to keep on thinking about ways, maybe with development of new methods to like less invasive methods to take like a biopsy or something that allows you to look at it.
Yeah.
It would be really, yeah, really add an extra important layer along the microbiological brain axis that we could measure in humans. So,
but there are things.
I don't have a solution, but maybe other people.
Yeah, but I mean, some stuff has been done. I mean, Carla sitting in the back, she has in Parkinson patients taken biopsies of those flashes and looked at enteric neurons whether they were affected.
Yeah, I know.
They didn't find major differences. No. But Carla also looked into functional dyspepsia patients, right? And there they found differences in the submucosal plexus.
GI patients, I mean, they typically need endoscopies, or you can still kind of convince them. I mean, people with like psychiatric or non-GI disorders, it becomes very hard, of course, to convince these people to just have an endoscopy for the purpose of research. And we're also really afraid that if that is mentioned as part of the protocol, people are going to be scared of and stuff like that. But maybe we should try to do it at least in like a smaller subgroup of patients or something.
Well, the thing is that the subtle defects in connectivity that I was talking about are probably going to be very hard to measure in those biopsies.
Yeah.
And I don't, I mean, I said there, I think that those subtle changes in the network can be a cause of GI disease. I didn't say that this is a cause of, I mean, CNS disorders. I won't.
Yeah, but of
say
course,
that.
then if you tie it into the
It could be
the whole story of your Nature paper that's going to come out, where you say, okay, how does it respond to different kind of nutrients or metabolites? I mean, that would be very interesting to be able to study in patients, obviously.
The question
we're looking at acetate, which is, I mean, which is a short chain fatty acid.
The question, so the reason why I always say, like, okay, the changes in the ENS could explain GI symptoms. I think it's logic that the same pathophysiological mechanism which kills or makes neurons die in Parkinson's disease patients or in Alzheimer's disease patients, those same mechanisms might happen in the gut and kill neurons there. And I mean, that's logic, right? Chicken or the egg or starting in the gut, that's so we don't have that evidence in these things.
But if we could study this relay function in some way, it would be nice, I think. But tough.
Yeah, maybe to reinforce sort of this discussion about the non-invasive ways. It's something that I would like to do also later, but Alessandro would lead. But I don't know if everyone attended, these smart ingestible pills and technology. Like, if you were to identify like 1 or 2 measures that you wanted to get and that are potentially, compatible with these biosensors, do you have something in mind that you could target in humans non-invasively?
I mean, those smart pills have. I mean, we're in the motility field already for a long time, and they've been used to map motility in patients already. I mean, if you could have like a robotic, I don't know, control over your smart pill to take a biopsy at the position along the gut where there is. Should be possible in the future, right? So, okay, we see an issue here.
The possibility to move the pill has been developed in standard.
With a camera and then take a biopsy at the position where you think there is a problem. That might be something to sample.
Localization is a problem that I think is a big problem, but it's something we should think about.
I definitely know that people at IMEC in Leuven, they every now and then they are also working on this kind of stuff like ingestible pills, and they're working on a version where you can not take a biopsy, but at least sample. Take a sample of the luminal contents in a place that you can decide on, and every now and then in their development process they come to us and like, what would you like to sample? So it's a good question. I would obviously like to sample short chain fatty acids because that's something we are focusing on, but you can think of many interesting things that you would want to sample, obviously. So these technologies are indeed evolving and developing and will probably allow us in a or relatively minimally invasive way to really sample things in the gut. Biopsies, I don't know whether that's already on the agenda. I'm not sure, but
what I can suggest also is that maybe something can be done directly in situ without sampling. I mean, we are developing, for example, tools for to monitor the cellular activity. So maybe in the neuronal case, it may be interesting to monitor cellular activity in terms of the calcium pump, any kind of pump which is regulating the electrical signals. And we are developing it without any patch clamp, so without any invasive way, just using an interface with the device, and we are doing in vitro. I don't know, but I can imagine that it can be theoretically done also in vivo directly. So you can apply something locally, so maybe through surgery, and keep monitoring this activity to any specific context you think is.
Of course, in mouse and rodents that is being done. I mean, people are moving into in vivo calcium imaging in the gut, so they create a window. A surgical window, and then you can go with a microscope close to the gut and measure activity. But of course, that's rodents again. You don't want to
with high resolution because you can be in contact, so it's identical to patch clamp. You get the same identical signal, so you can really distinguish between each segment related to each element.
So would that be an interface that you position somewhere stuck in the luminal side of the gut then, or how do I see that?
No, how to localize it?
Because that is an issue.
For sure, I can tell you that with cell we can do it. So maybe in future we can have also access to lab in the pill. That's what I mean.
Yeah.
You need to sample, but you can obtain the lab and take the pill, obviously, through complication.
So yeah, maybe something that I should also mention. I think I said we're interested in the ENS because it's very complex and it's more difficult than the CNS. That's all marketing, of course. But it's also difficult to study enteric nervous system for these kind of things because the gut moves.
Yeah, exactly.
Right. The brain sort of sits still.
Yeah, it's a moving target which makes things really.
And to then record activity of cells, I mean that's also your stomach, gastric, or gastric. How do you say it? GCGs?
Yeah, stimulated intestinal fluid.
No, the electrical activity that you mentioned.
Yeah.
Sorry.
Yeah, I was what you talking about.
It's moving all the time, but also these even in mice, if we do in vivo imaging of activity of neurons, we need to glue the gut to that optical needle because otherwise it just moves away. And you can do repeated measures like, we're going to look at the same cells the day after, but the gut has moved, so you don't find it back.
You cannot glue guts in humans, obviously. Yeah. So I mean, just an example. I was involved in one study where we were doing. I mean, this was in collaboration where we were doing both brain imaging and GI, MR-based GI imaging. To look at motility and water content and all of that kind of stuff with a group in the UK that specializes in this. I mean, this is tough analyzing these data because it's like moving like crazy, especially we were looking at responses to nutrients, so you get really a moving target. It's really hard.
So for the measurements that I showed in the introduction, the calcium imaging measurements, there we try to immobilize the gut. So by using pharmacological inhibitors of neuronal activity or for muscle contraction, but that of course you're interfering already with the physiology. The cells are there to get the gut moving. So if you want to record for them and then make the gut not move, you're already doing a lot of changes there. For that reason, I mean,
that's why probably if you are able to attach directly into the intestine, it will be a more stable and reliable. That's what we believe. Let's see.
I agree.
I really need to wrap now, but I will try to connect for the brainstorming.
All right.
Okay.
Thank
you all.
Bye.
Bye bye.
Okay.
Thank you.
Thank you again.